So, Dave Smith is the Vice President of Global Business Development at Lonza, and his presentation, Cell Therapy Commercialization, How Do We Get There? Dave.
Good afternoon. So today I'd like to really talk about cell therapy. Lonza has been doing contract manufacturing of cell therapy since 2002. We started the business at that time. We've expanded many times since that time, and we've become the experts really in allogeneic stem cells. The reason I wanted to bring this up is this is the base of all the work we're going to talk about today. Adult stem cells, whether MSCs, MAPCs, MPCs, or even if we go over to cancer vaccines, where we're using modified cancer vaccines, they all require large amounts of cells. And when people don't have a perspective of the size of the cell loads that we need. For simple diseases where we look at cancers, we look at osteoarthritis, we look at other diseases that are going to be treated with these stem cells, we're talking about incidents of tens, hundreds of thousands. We need to deliver somewhere between 100 million and 1 billion cells per dose. To give you a perspective of what that means, this means that the average very small 5,000 dose commercialization, you need 1 trillion cells per year. Put that in perspective, our average lot size right now is about 10 billion cells. So that means you have to produce almost 1,000 lots of cells a year. So we need to find ways to scale up. If we talk about very large indications, I can name some. Tissue Gene is working on right now on osteoarthritis. 200,000, 300,000 patients with dosing. The latest estimate is now somewhere over 1 quadrillion cells that will be required per year. So we need to do it a different way. So we actually started in 2002. Most people come to us in plates. So it's not unusual for this is the way we start. We moved over very quickly to a Nunc 10 stack. Very straightforward. To give you a perspective of where you can get on a 10 stack manufacturing, we started at about 5 billion cells. Through optimization, we can get to about 10 billion cells per lot. That's the best the industry could do through about 2012. 10 billion cells per lot. So you think about 1 trillion, 1 quadrillion cells, we're talking about a non-commercializable product. We moved very quickly over into some of the more high-density one, like the Corning HyperStack, where you're getting 36 to 120 layers per inch. Again, much higher density, much higher lot size. We can move this forward very quickly. But again, we hit a very maximum scale. We hit in a stage of about 100 billion cells. We can't go per lot. We cannot go any higher. So we've gone through these different stages throughout. Today, I'm going to focus on probably the first scale product that we can get and manufacture a commercially viable number of cells. So remember, as opposed to all the other technologies you're talking about, these have to be adherent because the cells cannot be in suspension. So you have to find a way to attach these cells to a bead, put them in a suspension culture, add more and more beads as you expand up because you need these things to go through passages and growing up. And in the end, you have to get the cells off the bead, alive and viable. This is a leap of technology that has been very strong. Because if you think about it, as I add beads and I add cells, what happens? You get these very large clumps of cells in the bioreactor. How do you get them? You have to use enzymes to break them up. When you put enough enzyme on them, you kill the cells. So now we're at a point where we had to come up with a unique bead structure that allowed us to get the cells off of the beads very quickly, very efficiently, without killing the cells. So our focus today is really going to be on microcarrier technology, where we scale these up to larger quantities. Wrong way. So, perspective. We're in 10 stacks. We can get up to about 10 billion cells. That only gives us 100 doses. Think about how many patients you can treat. The very smallest of all indications. You get to the very high density plastic, 2D, you'll see that 2D, basically a flat surface. You can get up to a maximum of 100 billion cells, about 500 doses per lot. Even a very simple indication, that means you're making more than 500 lots of product a year. That means QA has to release more than 500 lots a year. It means you have batch records that are truly 40 or 50 feet high in a year. Physically not possible. So that's why we're moving much more towards the bioreactor technology. But we had to do it looking at a couple things. One, we had to look at, can we produce enough product? Can we do it at a cost that's cheap enough? Because we're competing against monoclonals or other vaccines. And can we do it with a quality that's strong, that's comparable? I'm going to give you MSC data. Either it's from bone marrow or adipose. These are just the very standard models we use. We can do this whether it is MSCs or whether they're cancer vaccines. It's the same model and it's the same number of cells per dose. We can get today about 2 billion cells per liter. So so as we talk about this, we go into a 100-liter bioreactor. That means we can get doses, lot sizes of 200 billion. Our number of lots drop drastically already, and we can move forward with that technology. More importantly, as we scale up, we can go to a 200, maybe as high as a 500-liter bioreactor. Again, we can get up to maybe 1,000 doses per bioreactor. Remember, this is a unique technology because, except for the cancer lines, which are cell lines, these are primary cells, which means you can only double the cells so many times before they are done. So we hit a cap around between 250 and 500 liters in a bioreactor before we can expand any further. So again, commercially viable is what we're looking for. The growth curves. Also, instead of going to somewhere between a 14 and 21 day culture time, because we're doing these in a open suspension where you have agitation and flow going around the cells, we can actually get much faster growth than what was compared to in a 2D culture. Now you've moved from 14 to 21 days down to 9 to 10 days. We can turn these lots over much quicker. We can get the bioreactors up much faster and turn the total number of lots up over the period. It works whether we use MSCs or adipose or bone marrow derived. Also, we've had to make it we're contract manufacturer, which means everyone has their own love for which bioreactor they love. So while we use a finesse controller, we are using all different types of bioreactors and proving that you can use them generally comparable. I will tell you that some of the cells like certain bioreactors better than others just because of configuration of the bioreactors, configuration of the agitation. But in general, we can move them around. The trick is really around the controller. Just to give you a perspective, it takes about 600 cell factories. That means 600 plastic boxes that a technician has to pick up individually, change the media 3 times a week, passage them, and move them forward. So it is, to get that perspective, 600 cell boxes or 1 200-liter bioreactor. This is where we have to go to be commercially viable. Give you a perspective, just to get 1 trillion cells per year, which is not even a real indication, much smaller than an indication, we would have to have about 4,000 square feet of clean room just to do the upstream culturing. Doesn't count the downstream, doesn't count the fill finish, doesn't count the visual inspection. It will take that much space, whereas instead we can do 2 200-liter bioreactor trains, get the same amount of product, which a much higher quality than we were today. Also, cost of goods. This was actually early work we did. We have models around cost of goods. we can actually cut cost of goods. Today, we say conservatively 50%. It's probably with the new yields we're getting closer to 75%, where we can actually decrease the cost of goods overall and reach a cost of goods per dose that's below monoclonals. So for our clients, they can reach a point where they can now compete at a commercial level using bioreactors. The other thing that happens is cells die. I mean, that's the key. When you're harvesting cells, they die. You have a very limited time to harvest. This is a whole different concept. Remember, where everyone else we make drug substance, right? And then we move to drug product. This, drug product, drug substance, all the same thing. From the time you start the harvest, you have about 6 hours before you have to consolidate the batch. If you're doing large number of cell factories, you can't possibly do it. And once you start the process and you formulate, you have less than 3 hours to finish your fill finish, your formulation, your fill finish, and visual inspection. Because we're using so much plastic, you are getting plastic particles in these products. So we had to also find a way to limit the number of particles, much better with a bioreactor, but there's other ways we have to look at it. So your last harvest day is incredibly fast, large armies of people moving this thing through very, very quickly. Just to show you, we have been able at this point to show that when I move you from a 2D system, straight, flat surfaces, to a 3D system, all the surface markers that stay the same. We can move through and look at the standard MSC type markers. In every position, we can find that we can produce the exact same number, the exact same surface markers as we did in the past. Same when we look at CD45, CD19. We're also seeing it on some of the more complete markers that are included. This is where I was talking about. Particles are becoming the bane of cell therapy today. Picture plastic processing. You are doing closed processing. That means every time you change media, you have to weld a new bag onto the system. Every time you weld, you have particles. On top of that, when you're doing 600 plastic boxes, while you're making cell factories, it's a sterile weld, you actually will, again, induce particles into the system. Every single bag of media has particles in it. The bag production will leave particles in the system. By the end, you can actually do a visual inspection and have 10 to 30% of your product with visual particles in it. So if you did it no other process, that's what you can come back with. Now we're looking at different ways to do filtration of stem cells and other cells that are in group. Think about it. I can't push them through fast because shear will kill these cells. They're relatively large, so you can't push them through a micron filter. You can't push them through anything. So we had to develop techniques for high volume, low shear processing to decrease the number of particles that enter into the system. With this, we can decrease our number of particles down to a much more reasonable level. With that, we have been able to do it without losing viability of the cell. But again, this is required to happen within a very short period when you're doing the actual harvesting. So with this, we can get kind of a higher quality, less particles, more consistency. We can get very large quantities. And more importantly, we can get good quality systems because now instead of dealing with 60 feet of batch records a year, you're dealing with maybe 5 feet of batch records a year. That's much more manageable, except the quality guys will tell me it's not. And we can get to a point where we can get very comparable batches because as opposed to a cell factory, when you use a bioreactor, you can now control the oxygen, you control the nitrogen, you control the carbon dioxide, and control the system much tighter than you could with a cell factory. I'd also like to. Oh, I'm moving really quick through this. So we're going to be done. We're going to go. I'll go to the bar very quickly here. The last thing I really do is talk a little bit about changes in Lonza that's occurred very, very recently. It has been a very aggressive growth phase for us, and there's been a number of major announcements in the last couple weeks. The first one is, it's an unannounced, but we've started to move all of our commercial production to Portsmouth. We have our first major phase 3 commercial product that is going into Portsmouth. Portsmouth is the core of our biologic manufacture in the U.S. Anyone who knows it, it has 17 approved products. They know more about regulatory approvals than anyone. We're going to use that expertise in regulatory in Portsmouth. That will be our commercial site along with Singapore. With that, we can kind of leverage the expertise, because today no one's totally sure how the FDA, the PMDA, the EMEA will regulate manufacturing of these cells. So what we're going to do is we're leveraging off all the expertise through the years by putting it into Portsmouth and Singapore, which are our largest biologic sites. In addition, we will be moving our viral production to a building adjacent in Portsmouth. So again, we will have all of our biologics available. The commercial biologics will all be in Portsmouth. Also, Houston, there was a press release this week. We are expanding and building a new facility in Houston. Houston was an organization we bought in 2010. Expertise is on viral manufacturing. So they make viral vectors, and they make anything that has to do with a virus. So as you hear about the new technologies, CAR-T, anything that's being used on a viral vector to a human cell, that was all being done in Houston. We believe that from a contract manufacturer, we need to separate normal human cells from viral transfector or viral transduction, so that we get the highest quality, and we make sure that there's no potential for cross-contamination. The best way to do that, put them in different states. So we are moving forward with that. We are going to put them all in New Hampshire, but they will be not near each other. So again, we're focusing on that. The last thing that we've done is Japan. For anyone who knows what's going on in regenerative medicine, Japan is the hottest issue there is today. As everyone in pharmaceutical knows, you used to go to the U.S., you went to Europe, and then you went to Japan, because it was always the most conservative and the most difficult to move into. In regenerative medicine, the government has made a decision that regenerative medicine was safe and effective, and it will help on many of their... There's a lot of issues in Japan around the aging population and being able to afford healthcare costs. There was an agreement that regenerative medicine was a way to control their healthcare costs. They have changed the regulation so that with phase 2 data in key diseases, you can go onto the market with full approval in Japan. This is bringing forward a whole new marketplace. With that, Lonza felt we needed a strategic partner in Japan to take advantage of these regulations. After a lot of research, we chose Nikon. They have already moved forward with much of their technology on IPS identification, induced pluripotent stem cell identification and growth. We see them as a very good strategic partner. We closed the agreement with them about a month ago. It will be a Nikon facility, Nikon people. It will be Lonza systems. It will be our quality system. It will be our operating systems. And more importantly, these technicians will have been trained in the Lonza facilities. So we will quickly bring this facility up online, and it gives our current clients an opportunity to take advantage of these regulations without giving up the quality and the documentation that they currently are, the systems they currently are used to. So we will simply lift them up out of Portsmouth or Singapore, and we will move our clients into our facility in Japan. We see this as a huge advantage for our clients to take advantage of this kind of unique situation that's occurred in Japan. With that, thank you. Oh, any questions?